Really appreciate it.
Well, thank you all. It's delightful to be here, and I think it's exciting some emerging themes about spatial proteomics, open science, community efforts, and so I'll be touching on those. And another theme that I unfortunately won't touch on is phospho proteins, but that was exciting to see. And so, yeah, my new email address is here. And the work that I will show you, I did and originated at the NIH, but I'm continuing as a program consultant for HuBMAP and also with the IBEX imaging community. My focus is very narrow when it comes to applications. It's focused on multiplexed immunofluorescence and really highly multiplexed immunofluorescence. And this just shows you this rainbow collage that I made of the images, just the different anatomical structures and cell types. And this is always the why behind the how, that we would map these tissues, both normal and diseased. And in order to do so, we did this with antibodies. It was a very targeted approach using antibodies against these protein targets expressed in these tissues. And so the outline of the talk is to talk about multiplexed antibody-based imaging or targeted spatial proteomics, to talk about one method which I developed at the NIH, which is called IBEX. It's an open source imaging method that allows you to do high parameter, more than 65 antibodies in a single tissue section. And then two community efforts. Again, encouraged to see this open science movement here in the antibody validation community is one, a grassroots effort, and then two is a consortium-wide effort, including these organ mapping antibody panels. And so I show you this figure to be deliberately, it's very busy. It's meant to show you that because this field of spatial biology is rapidly evolving. And I just want to work on a couple of axes and not go through each individual method, but just that we can group these methods, and as Roberto did, about the resolution, the pixel size, if you're using an optical microscope, or for the sequencing-based arrays, the size and the design of the capture array. And so really with the light microscopy, we can get that subcellular resolution. We can also look at these methods based on the tissue thickness. Are we doing thin tissue sections that you'd get from a microtome, a traditional FFPE, or an area that the lab that I was in and was very near and dear to me is to expand to the 3D, that we want to look at the complex neurovasculature or neurons and vasculature and fibrosis, and we need to keep that in intact tissues. Another way of orienting these methods is the plex, or the number of analytes, if it's number of RNA molecules or protein targets for antibody-based assays, to the whole transcriptome and whole proteome. And so there's a lot of methods. It's constantly changing. And where you're seeing the evolution of these methods is improvements in resolution. I think also improvements in the depth of tissue that can be imaged, but we know that we need better affinity reagents to penetrate these tissue volumes. And then also improvements in the plex and combining multi-omics. And so just showing, yes, the Visium HD, now we have better resolution, if you will, with the new technology, and then also something that had been developed by Leica Microsystems, the company that I work for now, is the ability to do 15 plex in one go in thin and thick tissue samples using an advanced confocal instrument. And where I'll focus today, again, is the spatial proteomics. Those ones we have the exploratory, the deep visual proteomics, which combines laser microdissection and mass spectrometry. And then we also have these multiplex antibody-based imaging methods that can be broadly divided based on the format of the antibody. Are they using fluorophore labeled, DNA labeled, or metal labeled? And again, lots of different methods out there. And so as Roberto nicely showed, that this was method of the year. And this is why it's important. And you guys have been doing this for a lot longer than a lot of the spatial biology companies. And at its core, with, again, the targeted multiplex antibody-based imaging methods, at its core is great antibodies. And I was excited to see that from so many presenters this conference. And so, yeah, the method that I'll tell you about that was mentioned here that we developed is called IBEX. And again, there's many methods. And so we developed this in the Germain lab at the NIH, and there was two approaches: a manual approach, which was very tedious, and then an automated approach where we're using a fluidics device to deliver the antibodies in the dye inactivation solution. The general method, which is very similar to the comment presented by Zoe, and then also, I apologize, the representative from Biotechne, that we take a cocktail of antibodies. In our case, it's directly labeled fluorescent antibodies. We image, dye inactivate, and then repeat. And then you need to align those images into one composite using a common marker, a fiducial, usually nuclear marker. And this is just to show, okay, we've demonstrated this in mouse tissues as proof of concept, imaging anywhere from 20 to 41 in a single tissue section. We use this in the application area as a trained immunologist. So we were working on creating a human thymus atlas, and this was part of the consortium, the Human Cell Atlas. This was featured along with the whole paper package in November 2024. And with the Human Cell Atlas papers. And just showing you, this is an example of a thymus tissue section. We can see the beautiful cortex, we can see the medulla, and I'm just showing 4 of 44 parameters. And you probably can't see with the lights on, but just qualitatively, if you could see this image closer, and it's pseudo-colored with the reds and yellows marking the different keratins that are expressed differentially by these beautiful medullary thymic epithelial cells, you just see great diversity in this expression. We're bearing out seeing the pure yellow, oranges, magentas, all that, and we certainly can quantify that, and we did so in the paper. But just that these multiplex antibody-based imaging methods, qualitative and quantitative data, if done carefully, right? And then we can take this panel of 44 markers and use it to map these 7 anatomical structures, various subsets of T cells, B cells, looking, again, depending on the markers that you choose, cell state. And then in this particular question, we were interested in these thymic epithelial cells. And so that's why I really appreciated these panel designs, because you have to design the targets in advance, a priori. It's always chasing a question or a hypothesis, or if there's some research focus. And that was the case for this panel. And so that's just a brief synopsis of, okay, here's the method, here's how we used it. But one of the things I really want to drill home in the time that I have is the community approach. So when we first presented the method, we published the original method in 2020, and then a detailed protocol in 2022. But there's limitations with publication processes, and we heard that about, I think it was Johan trying to correct the record with the poor antibody. And so some of the limitations with the scientific publishing process is it's a static snapshot, right? And it's really old news at the time of publication. By the time you get reviews and so on, yeah, you're nodding your head. So my record for time from preprint to publication was 633 days. That nearly killed me, right? I mean, it was tough. And we report our successes, but not our failures, right? And failures, right? That what works. And either you're first or last, but we know that reproducibility is very important. So how do we capture that? And then that lab lore and those difficult steps may not be well described. How can I tell you that I jiggle it this way and then I soak off the cover slip? These things that really need to be captured with the video, right? And also what we appreciated as the community evolved was how experimental settings vary across time and space, that a reagent may be discontinued, or that it may not be shipped globally, or have restrictions there. And so our answer to this, and this answer that many of you have, is community, right? And this is a very grassroots effort, and I appreciated what Jim had said about the open science, open source software. So Ziv Yanov is not a biologist. He's a really talented software engineer, and he had helped with the image registration software. And he said, we're going to take the playbook that we use for simple ITK and create a knowledge base for multiplex tissue imaging. And the 3 facets are having a GitHub, which includes the repositories for data documentation and maintenance and scripts. We can take advantage of the discussion forum that's already a part of GitHub and an issue tracker for reporting problems. Then when we add new data, it could be a reagent resource, a protocol, a publication data set, or a link to data set. We do that with a pull request, and then it's automatically updated and reported out to a static website that's just generated through our GitHub scripts. And this is where we link out to reagents, protocols. Nothing is hosted really there. It just links to other material. And it's just a more user-friendly way of sourcing and searching the data, because we really call this a data set, not a website. And then when we've reached a significant level of knowledge, we do then a release to Zenodo, and that's a great way, as we see, the knowledge base. It gives us an authoritative, citable data set with a digital object identifier, and then we've made a point to incentivize people to contribute that everybody who contributes is named, and then that data set is downloadable, including all the reference images that we operate under the Creative Commons license, so other vendors could be using this to support. And so we publish this, and this is just the commercial. So this is in eLife. We also talk about the community, and if you scan the QR code, it take you to the static website. And this is what you would find there. Again, we want to share details about sample preparation. We want to have protocols, videos for multiplex tissue imaging. We have reagents that we recommend and also not recommend, and that's a very nuanced, and we'll cover that. And then we, again, want to encourage that you want to be a part of something, that you're not alone in the lab. And this especially resonates with junior scientists, that we want to celebrate your wins. We want to be able to support you with the discussion forum. And then we're really excited that we have people from all over the world, and this is a growing number contributing. And again, to incentivize this behavior, everybody who contributes, even if it's just one antibody, a protocol, a video showing some lab lore and images, reference images or data sets gets named on the Zenodo release. And so far to date, in the original IBEX manuscripts, we just reported human and mouse tissues, but we're happy that it's expanded, that people are using IBEX and multiplex tissue imaging on these experimental animal model systems, which, importantly, don't have a lot of reagents. So that's exciting too. If someone can validate reagents for this new model for aging, it's very valuable. And same thing again with the zebrafish. And then just to walk you through, there's a lot on the site, so it's not just reagents, but I think that's the thing that's the most interesting for everyone here, is that if we went to the static website and clicked on the reagent resources, then we have all these drop-down menus where we can search the data, and we can search by target species. And we can, the arrow just says to go to the right, and you can see that we have RIDs. We ask for that whenever possible. And then we can search for methods. So not just IBEX, but multiplex 2D, multiplex 3D. We want to expand this even greater. We have the tissue preservation method, because that certainly influences the performance of the antibody. I really like what Elena said, that we record what target tissue did you validate the antibody, because that will influence the performance of the antibody, along with the tissue state, the presence of a detergent in the labeling and blocking buffer, as well, of course, of your antigen retrieval conditions. And for IBEX, because we're doing cyclic rounds, it's really important to record the dye inactivation conditions. And then we say, do you recommend this reagent for that workflow? And if you click on the ORCID, then it would bring you to the reference data. We really, of course, appreciate if people share the reference images and notes about how it works. And then finally, one of the things that we work to create that we could do with the knowledge base is that we want to have a way of self-correction and reproducibility. So here you have a state where the original contributor is named by their ORCID. And then we have two people, they've been able to replicate that result by two individuals. And so that's rewarded. But we also want to capture the disagrees, right? And so we do have our first disagreement. That was a while back. We want to encourage, hey, that didn't work under those conditions, and then share that information. Okay, and I have until 12:15. So yeah, and then I'll go. Still just one more little, I have to go quickly. So the other community, which was a consortium-wide effort, and it's always been a part of a big team, and I recognize all the leaders here, Liz and Neil and Ellen, Diane, and Mike, and we've been operating for the last 5 years across 9 time zones with representatives from industry and academia and government, and some of you are collected in the audience here. In 2022, we shared a paper about multiplex antibody-based imaging and just offering, the overview of the field, but the problem and resources. In 2023, we offered a solution about specifically developing these highly multiplexed imaging panels. And we're working on a third paper about the impact and future of this asset. And just to jump quickly, is that you can trust me, if it's hard to validate one antibody, it's even more difficult to validate a series of antibodies that have to work. And so here, I'll just animate quickly, is because this is published and I can link you to the paper, is that we ask people to, okay, this organ mapping antibody panel is these OMAPs are collections of antibodies that map anatomical structures and cell types and tissues. You have the whole process of marker selection, antibody selection using CiteAb and other resources. Then Then you validate the antibodies using IHC as a reference, as well as positive and negative controls. Then importantly, you have to validate the full panel, the order of the antibodies, how does the panel, how does the antibody perform in cycle 6 versus cycle 1, all that, and then report that out. And we have a digital object identifier also for the OMAP. And to date, we have 30 different organ mapping antibody panels that cover 16 organs and tissues. Again, the emphasis is on healthy, 8 different spatial biology methods, and over 897 antibody validation reports. So for every antibody that's in a panel, we have a report that includes pictures and is published online. And so, yeah, this is what we've been able to map so far. All the different tissues, it's online. You could scan that QR code. It would take you to the OMAPs. We welcome entries from industry because that's important too. And so Miltenyi has contributed many. And the reference data set. And then what I think it's interesting is across these 897 antibodies, with the 30 different organ mapping antibody panels that people are using for different tissues, that you do see some emerging clones. And I think what's really important is that these clones are sourced from multiple vendors. And we certainly have we always encourage people to use monoclonals, especially recombinant monoclonals. And there's one polyclonal, which is used in 10 OMAPs for the lymphatics. And anyway, this is just a distribution of the vendors, that we have multiple vendors with contributing antibodies in these panels. And again, these are communities, so they're picking the antibodies for their targets. And then finally, I just will thank everyone. It's been a big group along the way. And again, it originated in the Germain Lab, and we have a community around the world for the IBEX method and HuBMAP. And I apologize that was fast, that's just the commercial, but if you have any questions, I'll be here. Yeah, thank you.